let's get into it. Let me just grab a quick sip. So inquiry question is, how does mutation introduce new alleles into the population? So module 6 is a really nice segue from module 5, where now when we look at mutations, firstly we're going back to our processes of replication, translation, transcription, and talking about mutations affecting those processes. Okay, here we are. So first question we have here is that we're going to look at is, how do mutagens operate, the different types of mutations. We're going to talk about somatic versus germline mutation, and then talk about the effects of mutations on coding versus non-coding DNA, and then look at causes of genetic variation, and finish off by talking about population genetics. So we're going to talk about mutations, gene flow, and genetic drift. So here we are. Inquiry question is, how does mutation introduce new alleles into a population? Remember, a mutation, firstly, is a permanent alteration to the nucleotide sequence of an organism's genome. This is where we are permanently altering someone's, or rather, natural circumstances or their own system leads to the alteration of someone's own nucleotide sequences, which then changes the proteins that they are able to make. Okay, so now we're going to look at, and here we go. Okay, now we're going to look at the significance of coding versus non-coding DNA sections of the DNA. So remember, not all of the DNA is coded for by proteins. Sorry, is coded not all of the DNA codes for proteins, my bad. So what we have here is that coding DNA, in fact, is very small. Like, it's a very small part of the overall DNA. Like, a very small part of DNA codes for genes. Very, very small. That's our coding DNA. The rest of it, we have junk DNA, we have enhancers and silencers, we have promoter sequences. That name might stand out because remember we talked about how in DNA replication and in transcription and translation we have promoter sequences that actually tell us when to or where to actually start these processes. We have introns, terminator sequences, and non-coding RNA as well. So here is the purpose of each. So genes are, remember, these mutations that occur to our genes, to a coding region, it can have an effect on the protein that is produced, and it can have a serious effect on cell function. Mutations that occur to the introns, they may have effect on splicing of genes. Remember how when we talked about post-translational splicing, we talked about how we take out the introns so that we are left with the extrons, coding DNA, so it can have an effect on the splicing of the gene. Promoters. Mutations to promoter region can actually inhibit the ability for a gene to be transcribed, because remember, those promoters are telling us where to actually start the process, where to actually start transcribing. So if there's an issue here, that will have an effect. Oops. Oh, my bad. Sorry. Let me just quickly forward. Oh, that's fast. Perfect, and my highlights are still there. Awesome. Alrighty, even though they're terribly done, sorry about that, but they're still there. So, we talked about promoters. Now again, terminators, same thing as promoters. Mutation to a terminator sequence can result in incorrect or elongated mRNA sequences because it doesn't know where to stop, so it can be incorrect and elongated. Mutations to enhancers and silencers, so these are the regulator regions. They actually they enhance certain genes and they silence certain genes when we don't need them. So if mutations occur in this region, it can actually lead to some genes being over or underexpressed, and that can lead to either too much protein being formed or too little protein being formed. Non-coding RNA is when mutations occur to non-coding RNA sequences, and that can result in alteration to important molecules. So non-coding RNA is important for those RNA molecules that we talked about in the processes of replication, transcription, and translations, like, you know, tRNA is an important one, right? So if there's a mutation to that, it can affect the function of like tRNA and mRNA. Finally, junk DNA. Mutations to junk DNA, as the name suggests, won't have a huge effect, and they probably won't matter. So we can see how mutations, for example, I'll change color here, mutations to obviously genes, to introns, to promoters, to enhancers and silencers, to terminators, and also to non-coding RNA, pretty much to most of them, and you know, almost all of them, mutations will have an effect. And that tells us that it's not the best, right? Like, it's not ideal to have those mutations.